Here my group is focused on studying host bacteria interaction and pathogenesis to try to develop host-directed therapeutics against these bacterial pathogens, particularly the drug-resistant bacteria like Staphylococcus aureus and Enterococcus faecalis.
We aim to elaborate host molecules that are targeted by these bacterial pathogens and to understand the mechanisms of how they cause infection in our system. With this vision, we are currently working with antimicrobial peptides that are naturally produced by our body and are also available from different natural resources like the marine microbiomes and the phytochemicals obtained from nature. We are trying to understand how these antimicrobial peptides modulate our innate immune system and provide protection against these bacterial pathogens. In the future, we aim to harness this knowledge to develop strategies to prevent the infection from these bacterial pathogens. Equipped with this knowledge, we will design host-directed therapeutics and novel antibacterials, particularly against the drug-resistant bacterial pathogens like Staphylococcus aureus and Enterococcus faecalis. As a long-term goal, we see ourselves translating the knowledge we gain about host bacterial pathogen interaction into principles to develop host-directed preventives and adjunct therapeutics, particularly for fighting the silent pandemic known as antimicrobial resistance or AMR. As a future direction in our lab, we aim to identify newer pathogenic bacteria that might be available in the natural environment, identify virulence factors produced from these bacteria, and try to find out the host targets that are being affected by these bacterial pathogens to cause infection and create mortality and morbidity in the patient population.
My study is upon role of host antimicrobial peptide in shaping immune response during bacterial infection, particularly Staphylococcus aureus, which is a life-threatening bacteria, second position of high priority list by WHO. Like Streptococcus pyogenes, Staphylococcus aureus also targets LL37 triggered immune response, which resulted into persistence of the bacteria and caused diseases such as endocarditis.
Myself, Dr. Shubhendu Kumar Chatterjee. I am validating marine extracts and isolates for their antimicrobial activity against resistant pathogens and assessing their potential as immunomodulatory agents that can modulate inflammatory responses in skin models. Beyond therapeutic applications, I am also looking for industrial potential of marine microbes, especially their role in biodegradation. Some of our strains show promise in breaking down hydrocarbons like petrol, crude oil, and even synthetic plastics, offering eco-friendly solutions to pressing environmental challenges. Through this multidisciplinary approach, we aim to bridge marine biotechnology with human health and environmental sustainability.